Now this is my second YouTube video. I don't know how it's gonna turn out, but I really want to share my ideas in a different format, so here we go. Alright, so given that introduction, I want to introduce you now to this video itself, in which I'll be explaining what it takes to be a biohacker. This sounds absolutely nuts. Like, what is biohacking in the first place? Well, I described it as doing science outside of prestigious institutions. Like, anyone can cook, you know that quote by the chef in that Pixar movie? Well, I think biohacking is about the same thing. I think science should be a right, something that everyone can do, because it's going to be essential in the 21st century. Especially the next decades are gonna be full of life. What do I mean by this? Well, that the intersection between biology and technology is going to rule. If you've read some of my articles in my Medium blog, like Biology in the 21st Century or Synthetic Biology in a Nutshell, you may already know that I'm very passionate about this topic. That's why I decided to do this video. Because just after I had discovered CRISPR, I got a little disappointed. I discovered that it was also one of the things that people used to do only if they had enough money and enough knowledge. But then I discovered the Odin. The Odin is a company founded by Josiah Zayner, one of the only biohackers I actually know of, and the coolest one of them actually, because he really has this idea of spreading science around the world with people like me who are just teenagers and are curious people who want to learn about these technologies and who probably don't have the resources or the credentials to do science like having hands on CRISPR. Well, what they do at the Odin is that they sell kits such as genetic engineering kits that come in a box and they have all the necessary things so you and me can do biohacking at home without need of anything else. So yes, in this video we will be going through the process of making bacteria glow. This idea is also crazy. And the first question that you may be asking yourself is, why would you make bacteria glow in the first place? Why? Well, it actually turns out that scientists have been doing this since a very long time ago, but they use it as a technique within a much broader and longer and much more complicated process in order to know basically if their experiments worked. Now in this case, we're gonna be doing it just because of fun, just for the sake of learning. But it's important for you to know that this is not only about biohacking, it's also a technique that is used in academia and in the industry. Another question that's important to answer before actually getting into the experiment is why using bacteria? Why not yeast cells? Why not my cat cells? Why not my own cells? Which is actually possible. Well, the reason is because bacteria are kind of the simplest organisms that we know of. Reason number two is because they multiply really fast. What this allows us to do is to just make things more efficient, to make them faster. Next thing is, how are we actually gonna make it glow? So yes, I'm sorry to say this, bacteria don't normally glow. They just don't like it. They maybe are not made for that. But the way we are going to do it is by doing genetic engineering. What this means is that we are gonna insert a piece of DNA called a plasmid. A plasmid is basically DNA in a circle form into the bacteria. So our bacteria are gonna glow green basically because we are going to insert this plasmid with a gene that codes for a protein that's gonna make it glow. Now the third or fourth question, I'm not sure. How are we actually gonna insert that plasmid into bacteria? How does that actually look like? Well, you're actually gonna see how that looks when I get into the experiment part, but for now what I can say is that we are gonna use something called a transformation mix. This basically consists of calcium chloride, and what this is gonna do is it's gonna interact with the cell membrane, which is its kind of protective layer, and it's gonna tell it like, hey, we need this plasmid to pass into the cell. You know, they don't actually talk, but it's like a chemical language, and basically it opens like a space for the plasmid to get into the cell, into the bacteria cell, and that's how the magic happens. It's finally here, the thing you've been waiting for, the actual CRISPR kit. First things first, we need to take into account sterilization. We can spread some normal alcohol around the surfaces that we're gonna work on, as well as our gloves and hands. We measure the water that we're gonna use to make the plates. Remember, the houses for our bacteria, the food. We put it into a normal glass bottle. And then we add the LB agar media. Remember not to use the Albican agar media, which is the poisonous one for bacteria. Then we can shake it off. Put the lid back on. Do not tighten it a lot. And now we can put that into the microwave. Just a normal microwave. Yes, the one you use in your kitchen. I said 20 seconds, but it really depends on your microwave. Make sure we have a homogeneous mix. We prepare the actual plates, and we can pour the mix into those plates. And done. Now we're gonna let them dry for a while. Now next step is liquidizing bacteria. Bacteria comes in a freeze-dried way, so you're gonna be storing it in the fridge until you are gonna use it. Using the transfer pipette, you put some water into that container. And again, this is an example of how you shouldn't make plates. This is a good example of a nice bacteria house and food. Awesome. Next thing we're gonna do is put the bacteria back home. Let them enjoy the food and their new house. Using an inoculation loop, you're going to take some bacteria from our tube and spread it around the plate, gently. Let's take some more bacteria, put it in the plate. Nice. And make sure to keep that just in case you want to use that bacteria again. Put the lid back on. Awesome. And after some hours, we can now get bacteria growing. Isn't it awesome? Like you are actually seeing life in front of your eyes. Next step is scraping it out of a plate. Gently again. We don't want to break that agar. Using an inoculation loop. Yeah, we actually have a lot of it. Great. We are going to put it into our transformation mix. So yes, that tube that I'm just holding there is called the transformation mix. And we are actually gonna mix it. Now it's time for the interesting thing. Let's work with DNA, with the GFP plasmid. That was previously on the fridge, so we take it out of there. And using a transfer pipette, we transfer the bacteria in the transformation mix tube that we just worked with. We take it from one tube to the other one. There we go. We actually put all of it inside the GFP tube. We put it inside our fridge, not the freezer, the fridge. Bye. And we wait there for 30 minutes. Then we get a glass with hot water and incubate the tube inside there for 30 seconds. Now the next step is going to be giving bacteria some food. We are going to use the microcentrifuge tube that contains the LB agar, mix it with water, and put it inside our bacteria GFP plasmid tube, which contains the bacteria that have already been transformed and that already contain the DNA. Next, we take the Albican agar plates out of the fridge These are basically made the same way as the other plates. Remember, they contain bacteria venom. This way we will be able to select for the bacteria that we're able to get the plasmid. We use our transfer pipette to put the bacteria inside that plate. And we are almost done with our experiment. Next thing we do is we spread the bacteria around the plate with the spreader. Finally, we just need to put the lid back on, and don't be like me. Wait a little bit of time before putting the lid back on, and if things went right, you're gonna be able to see bacteria glowing. Did get bacteria, but the thing is that it was bacteria that we didn't want to grow. What this means is that we got our plates contaminated, and we won't be able to finish the experiment. Nevertheless, this was a very valuable experience. I really hope you've enjoyed this experiment as much as I did. I hope you have a wonderful day, and I'll see you in the next one.